everybody. Welcome to another episode of The Living Algorithm. So, Sushant, good morning. How are you doing today?
Good morning, very well.
Folks, today our discussion is going to be around immunotherapy. So, basically, immunotherapy was supposed to change everything. For the first time, weren't just poisoning cancer cells or cutting them out, but we were teaching the body's own immune system to hunt them down. And in some patients, it works spectacularly. But CAR T therapies have turned certain blood cancers into chronic or even curable disease. However, there is some uncomfortable truth that most people never hear. For the majority of patients with solid tumors, immunotherapy simply doesn't work. Response rates for the most common checkpoint inhibitors hover between 15 to 30% overall, and in cancers like pancreatic, prostate, glioblastoma, or even colorectal cancers, the numbers are far lower. So, why does a treatment that can produce miraculous durable remissions fail so often? Is this tumor's clever defense, the hostile neighborhood around cancer, the patient's own immune system, or something we don't fully understand? So, today we are going to unpack the real reasons immunotherapy succeeds in some people and fails in most, and what therapeutic approaches going forward are applicable in cancer. So, Sushant, my first question to you is when most people hear immunotherapy, many assume that it is a universal breakthrough, right? So, how accurate is that perception versus the actual numbers that we see in the clinic?
We have to understand the entire reprogramming and the epigenetic programming which happens when, immunotherapy is taken place or when we are giving bouts of immunotherapy. So essentially, before that, we have to understand that, a normal way of, and that is a normal way that our body produces cancer cells every single minute, and there are immune cells, immune surveillance, which happens. So it's like a police surveillance. The immune cells go and look out for cancer cells, and then they, express specific inflammatory cytokines to actually kill them, and that's a normal process, right? But when they are really cancer cells are growing, right? When they are in, when, as we discussed, there's a dysfunctional mitochondria, and cancer cells start fermenting glucose and glutamine for their survival and growth, right? So they grow, and they a lot of persistent antigen being formed, right? Tumor antigen, and then they also take up this high amount of glucose, which deprive the immune cells of the fuel which is required to perform their essential functions, right? To kill them, to kill these cancer cells. And again, there also a situation of chronic hypoxia, which is for people, maybe laymanish, it's like low oxygen environment, right? Which is conducive for cancer cells, and no cell, healthy cell cannot work, it cannot perform, cannot produce, transform energy without oxygen. So essentially, when these three factors are there, the cancer cell itself suppresses the proliferation of immune cells. Immune cells have effective functions, it loses the effective functions. This suppresses the inflammatory cytokines like TNF alpha, which are required to suppress cancer cells. Then the most important part where your immunotherapy comes into picture is a specific protein called PD-1. This is programmed death
Hmm.
1. This is expressed by immune cells, right? And it's a normal immune response. So basically, this particular there's a modulation of this protein which is required to actually tame down or lower the activity of immune cells, right? It is meant to maintain immune homeostasis, so that there's low immune activity when they attack the insults or whatever, the risk which body is experiencing, and then this is a normal way, right, of and so this PD-1 actually regulates the immune responses so that it does not lead to autoimmune damages, right? So again, PD-1 is sort of, in simple words, it lowers the T cells, immune T cells. So we're talking of the CD8 T cells, CD4 T cells, right? All these are immune cells which are required to kill cancer cells. So again, this lowers the activity of PD-1, and then these cancer cells actually, what they do is they express a specific receptors PDL1 on their surface, and that binds to PD-1, and that increases the expression of PD-1. So it lowers the activity of immune cells, right? So probably they prevent the immune cells from attacking them, right? That is one way. Other way, which we would discuss in the next podcast. But it is important that there are certain microbes which reside in these tumors, and these tumors express these microbes on their surface. So probably we know all that, it is your microbiome that trains your immune cells. So probably that tells immune cells that there's nothing to worry, there's nothing bad going on, so immune cells do not attack. That is another way. But essentially, cancer cells exhaust T cells, right? By depleting nutrient by creating an hypoxic environment, by actually driving the expression of specific exhaustion genes, tumor the T cells exhaustion genes, right? By inhibiting inflammatory cytokines, by suppressing their proliferation, right? So all these actions are taken by your cancer cells to suppress the immune cells activity and to exhaust T cells, right? Then also they also reprogram, methionine and tryptophan metabolism, which are the amino acid which we know, and this depletion of these methionine and tryptophan is something which suppresses the proliferation of T cells and make them exhaust, right? Then this cancer cell can also reprogram methionine into S-adenosylmethionine, which suppresses AMPK pathway, which drives the exhaustion of T cells, right? That alters the genetic profile, epigenetic modification that promotes the exhaustion of T cells. So there are a lot of these things, and even upregulate PD-1 inhibitors, right? So essentially, this reprogramming of methionine by your cancer cells can further, suppress the immune cells, right? And then again similarly, and another important aspect is, this also impact the mitochondria of T cells, right? So by the fact that one is obviously the tumor antigen response, other is nutrient depletion, right? So essentially, these PD-1 overexpression, downregulates metabolic pathways, right? Which immune cell may resort to transform energy to kill those cancer cells. So when there's high expression of PD-1 by overexpression of PDL-1, there is something called, this in fact, what it does is, it actually drives the metabolic reprogramming. It restricts the glucose uptake. It actually impairs the oxidative phosphorylation, right? It lowers energy production, right? And it leads to exhaustion of T cells and suppression of proliferation of T cells, right? So by all these methods, these actually, I'm not talking about, still I've not spoken about the immunotherapy, right? So these all are the way how metabolically and by epigenetic modification, right, your cancer cells can exhaust T cells, right? And specifically most important part is the mitochondria. So again, mitochondria, these by fermenting glucose and glutamine, these we have discussed during the last talk also, that these cancer cells produce lactic acid and succinic acid, and that makes T cells unable to perform their job, unable to to proliferate and kill cancer cells, right? And then nutrition depletion and antigen, all these leads to dysfunction of the mitochondria. There's downregulation of PGC1 alpha, which is required for mitochondrial biogenesis. So there's low mitochondrial quantity and function, and this actually, further impacts cellular metabolism and even upregulate, exhaustion promoting genes, right? And there's low ATP production, there's loss of membrane protection, there's high ROS, right? All these drives the metabolic disturbances and defective mitophagy, right? Which is again, mitophagy is, we have discussed it, mitophagy is killing off, getting rid of dysfunctional mitochondria. So because of this, there's a defective mitochondria which leads to metabolic disturbances, right? Which leads to decline in function of again T cells, right? So this is way how cancer cells actually, could reprogram their environment, tumor microenvironment to prevent T cells from attacking them, right? Now there are immunotherapies like the PD-1 therapies and all those therapies which which are meant to down-regulate the expression of PD-1, right? Which so that improve the activity of T cells. And again, CAR T therapy, which you mentioned in the beginning, right? Again, CAR T therapy is nothing but T cells are taken out and it is re-engineered so that it can be programmed to kill cancer cells. But again, all these needs functional mitochondria, right? So any T cell, any cell needs mitochondria. So we all know that all cells inside our body except the red blood cells have mitochondria, right? Functional mitochondria. And there are thousands of mitochondria in every cell, right? So essentially, even the CAR T therapy could only work when the mitochondria is functional, right? So essentially, all these factors have a role to play, and when your cancer cells is overpowering, is making your T cell exhausted or suppressing its proliferative activity, it could make these immunotherapies, ICI, immune checkpoint inhibitors, what we have is, right, ineffective, right? And again,
Hmm.
this is and again there are different these T cells. There's a lot of heterogeneity which again depends upon what kind of immunotherapy you are using, whether they are being effective, whether your microbiome, which we're going to discuss now, is how your microbiome could influence the effectiveness or render the PD-1 therapies ineffective, right?
Right, right. So before we go to the microbiome part, a small question which is very important, of course. How important is the tumor microenvironment, the surrounding cells, the blood vessels, and the chemical signals in
Yeah, so
determining the
I'm
success
I'm
or
coming into
failure?
that also. That's a part of
Yeah,
microbiome. So essentially, when
yeah.
cancer cells need to grow, they promote angiogenesis, which is the formation of new blood cells, right? Because
Hmm.
they need that thing to grow. They need nutrient to grow, right? Because you need nutrients, glucose and glutamine to grow. So essentially, they make new blood vessels, right? Which
Right.
actually help them grow, right? So again, this does not lead to metastatic cancer, because what happens is when there's a metastatic cancer happens when they are, there's a lot of these, they're doing inflammation, this cancer cells, and then immune cells can sense it, and immune cells are genetically programmed to enter and exit any cell. So when immune cells goes to the site of injury to kill these cancer cells, they actually fuse with these cancer cells, right, in a bid to kill them, but they themselves become rogue. They themselves become they acquire the properties of cancer cell, which is fermentation properties and proliferation properties of fermenting glucose and glutamine, because cancer cells have dysfunctional mitochondria. They cannot transform energy using oxygen, which is a normal way, and again they resort to fermentation pre-symbiotic era pathways, which is fermenting in the without oxygen, fermenting glucose and glutamine, which are two fuels, and they produce lactic acid and succinic acid that creates a shield to protect cancer cells. And again, these makes your T cells also not function properly, right? So essentially, the immune cells and macrophages, so basically macrophages can fuse with these cancer cells, and they themselves become rogue. And since they are programmed to enter exit any cell, so this leads to metastatic cancer, right? So essentially, angiogenesis lead to growth of cancer cells, but metastatic cancer is again a function of how your macrophages become rogue, by fusing with these cells, and they start spreading everywhere, right?
I get it now. So next very important question is that how does the gut microbiome or the patient's overall immune health influence whether this therapy succeeds or not?
Okay, so let us discuss, divide this into two parts, right? So
Yeah.
one is how the role of these microbiomes. So again, I'm not talking about the presence of a bacteria. I'm talking
Hmm.
about the metabolites these microbes produce, right? So one thing is these A lot of these during cancer, there's a lot of stress which could disrupt your oral microbiome, which has an impact of producing inflammatory cascade, which further fuels cancer, which is very well seen in the head and neck cancer, right? Uh, but in terms of the certain metabolites, so gut microbiome could produce short chain fatty acids like butyrate, or or maybe which is another tryptophan metabolite called indole acetic acid, right? All these are also set to regulate your blood sugar response. So again, these are directly connected with the impact of you know uh, the PD-1 therapies, immunotherapies. So essentially, butyrate can enhance the effectiveness of PD-1 therapies, right, to kill cancer cells. So if your gut microbiome is producing enough butyrate, I'm not saying you have a bacteria to produce, if they are actually expressing genes to produce butyrate, right, which is also a chemical chaperone that can kill toxins, right? We all know that it has role to play in longevity, in inhibiting inflammation, in making your gut more strong, so your gap, closing the gap junctions, and a lot of other beneficial factor. But this also makes the PD-1 therapy effective, right, by inhibiting inflammation, right, by inhibiting oxidative stress, by clearing toxins, right? um, okay, so that is one thing, right? And again, it can down regulate PD-1 as well, right? Which we all know that PD-1 is a protein that I've discussed earlier, that it lowers the function of T cells. So by inhibiting or by downregulating PD-1, it could help T cell function effectively, or it could improve the effectiveness of PD-1 therapy. PD-1 therapies are meant to downregulate PD-1, right? Or downregulate PD-L1, which is again the receptor expressed on the surface of cancer cells, right? So that is one. Other is indole acetic acid, which is again your gut microbiome ferments tryptophan, which is an amino acid, and produces indole acetic acid. And indole acetic acid, actually, what it does, it restores the function of CD8 T cells, right? It actually, have an inter-tumor impact, and it could even reverse there are certain gut bacteria that could make the PD-1 therapies ineffective. When you have gut dysbiosis, these therapies, one of the major reasons why these therapies are ineffective is gut dysbiosis. So there are certain gut bacteria, right, that could render the PD-1 therapies ineffective. They are resistant to these therapies, right? So this indole acetic acid can reverse actually this resistance caused by these bacteria to PD-1 therapies, right? So essentially, that is how these two metabolites are very super important, right? For improving the effectiveness of PD-1 therapy, or restoring the function of T cells, which is required to kill cancer cells, right? And then, another important thing is bile acid metabolism, right? So bile acid metabolism is a two-edged sword. So bile acid metabolism can help improve the effectiveness of PD-1 therapy, right? It can promote, or rather, it can actually promote the immune-mediated tumor control, right? Or if there's a dysregulated bile acid metabolism, which is generally caused by gut microbial dysbiosis, when there's too much of secondary bile acid, it could cause oxidative stress and endoplasmic reticulum stress, which could make these therapies ineffective, or which could exhaust T cells, right? So this is from a point of view of making or not making these therapies effective, right? By so fixing your gut microbiome and understanding what metabolites they are secreting, or altering your nutrition choices, that could actually stimulate production of these metabolites, could really help, right, in improving the effectiveness of immunotherapy. Unfortunately, currently, the cancer practice does not, or maybe any doctor does not appreciate the role of microbiome, right, as such. So this is a
Yeah,
bigger
yeah,
problem.
that needs a wide education, and they need to understand the mechanism. Yeah.
Yeah,
So, okay, now going
that
forward,
is,
what,
yeah, yeah.
yeah, so looking forward, let's say, what kind of combination strategies or new approaches look most promising, for overcoming this resistance towards the therapies?
So one thing you have to understand is that So I'm talking from the therapy's perspective. I'm not talking about the bioelectricity. So I'm not going to touch about nitric oxide this in this session, but I'm going to talk about another metabolite, another postbiotic, which is produced by gut microbiome, which is urolithin A, right?
Right.
And urolithin A is something your gut microbiome ferment elagitanins, ellagic acid in various foods like pomegranate, berries, strawberries, blackberries, pecans, walnuts, right? All these have a lot of these ellagic acid and elagitanins that can be converted into urolithin A. Now, urolithin A is a super molecule. It plays a very important role. It modulates your cell arrest cycle. It promotes apoptosis of cancer cells, so it can kill cancer cells. It inhibits angiogenesis in the tumor microenvironment. So as I mentioned, it
Mm.
angiogenesis again is a way of for cancer cells to make more blood vessels to grow. So it prevents
Mm.
that. It inhibits inflammation. It inhibits
Mm.
expression of matrix material protease 9 MMP9, which is said to cause inflammation, oxidative stress, which we have discussed in various talks, right? It
Mm.
also stimulates mitophagy and autophagy. So mitophagy is
Oh.
getting rid of dysfunctional mitochondria. So probably if T cells are not working or CAR T therapy is not working, is because of dysfunctional mitochondria. So urolithin
Mm.
A, either via fixing your gut microbiome or taking postbiotic, urolithin A postbiotic could help get rid of dysfunctional mitochondria, and it could also stimulate PGC1 alpha, which could make more mitochondria, new mitochondria per cell, and this can promote, the energy production via oxygen, right, which could help T cells kill those cancer cells, right? So, and again, these can also modulate specific signaling pathways like P53 mutations, FOXO1, or AKT1, or maybe NF. It downregulates NF kappa B and TNF alpha and a lot of these things, right? And then it also prevents tumor growth and invasion, right? So
Mm.
essentially, this particular molecule of postbiotic is should be part of every cancer therapy. It should be combined with cancer therapies to make it effective. So first thing is obviously you have to fix your gut microbiome. Please do not think that a I've seen there are companies who are selling at 5,000, 8,000 rupees gut microbiome test, which they just identify a bacteria. They cannot measure the actual metabolites being produced and how those metabolites impact the host cells, as I've discussed, or impact the immunotherapy. So essentially, these companies are just telling about, you have some producers, you have a potential to produce. They're not telling, in fact, actually, you're producing or not producing, right? So essentially, you need to understand the metabolomics, which is metabolites being produced by your microbiome, and fixing your gut microbiome, oral microbiome, because your oral microbiome directly impacts your gut microbiome, right? So oral
Mm.
microbiome metabolites or the translocation of oral bacteria could impact the gut microbial dysbiosis, right? And again, these specific metabolites, along with postbiotics such as urolithin A, when combined with immunotherapies and other therapies, could make cancer therapies effective, could improve the 5-year survival rate. And then the focus should be on fixing your microbiome and mitochondria, right? So that should be the
Mm.
right approach when it comes to fixing your improving the effectiveness of cancer treatments, right?
That's true. That totally makes sense. So, again, another question that I have is that are we getting better at predicting who will actually benefit before we start the treatment? Is there any sort of a predicting mechanism or a test
Yeah,
that
we do,
can
we
tell us that?
do, we do have independent of a cancer test, which most of us still we do not have any cancer tests as of now. Most of the companies are focusing on late stage random genetic mutations, right, or identifying gene expression products, which are again downstream effect of dysfunction in the mitochondria, right? So
Mm.
certain predictive biomarkers which could be looked at. I'm not talking about the blood biomarkers. So one is obviously looking at butyrate production, looking at bile acid
Mm.
production, look at indoor
Mm.
acetic acid production, right?
Mm.
Looking at lactic acid production, succinic acid production, looking at antioxidant production, urethane A production, right?
Mm.
So these all can tell if these pathways are dysregulated or
mm,
downregulated, or maybe lactic acid succinylase is upregulated, which clearly means
mm,
that you have an hypoxic environment, and your
mm,
cancer cells are actually fermenting it, or probably there
mm,
is a lot of fermentation, even though there is no cancer cell, there are a lot of fermentation going on. That's a temporary way of generating energy. So probably just think that when we do exercise and when we do a lot of high intensity exercise, and we do not have that kind of oxygen to support our activity. So there is the body temporarily resort to the lactate production, right? Essentially,
mm,
that again is used to convert into energy. So again, lactic acid, if lactic acid and succinic acid are upregulated, these are one of the early risk factors for cancer, right?
mm,
Then down regulation of, or lower expression of genes, that microbial genes that could lead to transformation of ellagic acid into urethane A is again another risk factor. Low butyrate production, right, which again could be caused by glyphosate, molds, ultra processed foods, maybe consuming a high
mm,
protein diet, right? Protein fermentation, as again another signal. For instance, in oral cancer, hydrogen sulfide and methyl mercaptan production are one of the key pro-inflammatory factors that could lead to dysfunctional mitochondria, that could downregulate superoxide dismutase, which is required to clear the toxins, which is required to overcome oxidative stress, right? So all these are molecular pathways which has to be looked into, right?
yeah, that's correct. That's absolutely right. So before we conclude this podcast today, I'd like to
Mm-hmm.
ask you a question from the patient's perspective. So for
Mm-hmm.
patients who actually respond to immunotherapy, what does
Mm-hmm.
long-term success actually look like, and what trade-offs or side effects they still might face?
So yeah, so these therapies have side effects. These therapies, basically, these toxic therapies could have a collateral damage, because when you are targeting certain gene expression products, certain driver mutations, right, which can also be found in
Hmm.
perfectly healthy cells, right? So it could lead to kidney issues and liver issues and a lot of other things, right? And
Yeah.
then also. Even the therapies have become effective, you have to fix your microbiome. Please focus on
Hmm.
fixing your gut and oral microbiome, right? That
The
is very important.
microenvironment around, also needs to be fixed. The the environment around the cancer cells,
And
isn't?
then last time we have discussed that you have to fix your oral health, because that's the starting point of most of the
Yes.
cancers, right? So fix your oral health, fix your microbiome, do not fall for these influencer diet regimes, these diet regimes based on your variables or your blood test, right? These are
Yeah, that's
very,
correct.
very,
That's
very
correct.
minuscule part of your overall investigation into your biology, right?
Mm.
So that is something which has to be considered, right? Uh,
So, Sushant, any last words would you like to add for the oncologists, for the researchers, for those working in the cancer treatments?
so please read papers. There are a lot of research which is around it, which clearly establishes the fact that it is you know break down in the energetic process of the mitochondria that causes cancer, right? And please understand, this is a common sense, that your mitochondria transforms energy. Without energy, you cannot survive, right? You're
Mm,
breathing, you're thinking, you're eating, it all needs energy, right? So, and mitochondria, since it controls the apoptotic signaling, it controls whether cells survive, die, grow, proliferate, right? So when
mm,
that
mm,
signal is gone, when that control is gone, that's how cancer cells grow, right? So please understand the role of mitochondria in the onset of
mm,
cancer, right?
mm,
Genes, mutations, or the damage to the damage to DNA, RNA, protein are just downstream effect of the defect in the mitochondria,
mm,
when the mitochondria become corrupted, right?
mm,
And then another thing which is most important is, please fix your microbiome. Do not resort to this low cost microbiome tests which have zero understanding of the metabolites and how those metabolites communicate with your cells and with your mitochondria. So essentially, these gut microbiome release the metabolites. These metabolites are released in the bloodstream and they travel to your mitochondria and they interact with the mitochondria. And based on the level of these metabolites, they could either you know promote a better ion channel regulation, and they can better energy transformation, or it could lead to high energy resistance, which again could lead to production of reactive oxygen species, which could damage your DNA, RNA, and protein that leads to your cancer proliferation. And again, another angle to look at it is, there's stress costs, right? So essentially,
Hmm.
these metabolites are nothing but stress costs. Of probably, when there's too much of stress, there's a reallocation of energy from other growth, maintenance, repair functions to these responding to these stressors. And then when this happens, there is a lot of increased energy consumption, increased energy expenditure, right? And when there's an increase in energy expenditure at one point of time, your cells cannot use oxygen to transform energy, and they resort to fermentation model. So when there's too much of energy expenditure, there's less energy consumption, more energy expenditure. So you don't have any energy to use oxidase phosphorylation to transform ATP, right? And then they resort to your fermentation pathways, right? So essentially, that is another angle of how you look at cancer. So please study these papers. Please listen to you know Thomas A. Fried, Professor Thomas A. Fried, whom we have also invited for a talk last year, right? And he has more research now. How do you prevent cancer? Please stop feeding sugary foods to cancer patients just because that there's a cancer cachexia happening, where your cancers eat up the muscles and bones of muscles and bones to basically to convert into sugar for their growth. So essentially, you know feeding sugar is not going to help. Please resort to therapeutic ketosis, where you do not cancer cells cannot ferment fatty acid, right? So if you do fasting and a lot of these ketosis alongside your chemotherapies and other therapies, it can really help cancer patients.
That's really
So
a great
my
message that
only
you've
only
given.
advice is learn, read. Please read. Do not assume.
Yeah, that's true. I mean, we do need to go through a lot of research happening around the current
Mm,
breakthrough therapies that are coming up. So,
mm.
Sushant, this was a very interesting discussion for both the patients and the oncology perspective. So, thank you for sharing this important aspect and important research that you found out. So, folks, thank you again for giving us your love, and those of all you've been following the living algorithm. Thank you so much for listening. And in the end, I'd just like to say that, immunotherapy remains one of the most exciting tools we have. But understanding why it fails is the only way we will make it succeed for more people, right?
Yeah.
Isn't
So
that right, Sushant?
yeah. So just want to add, and from a layman perspective, please understand that immunotherapy is meant to make immune cells function appropriately to kill cancer cells, right?
Yes.
And they can only function when they have energy, when they can transform energy. And for that, they need to have a functional mitochondria. So whether you're resorting to PD-1 therapies or CAR-T therapies, you have to fix the mitochondria. And the best way to fix mitochondria is fixing your gut microbiome, because urethane plays a very important role in triggering autophagy and PGC1 alpha. So your natriuretic oxide can also stimulate PGC1 alpha, which is making more mitochondria, healthy mitochondria, right? So all these have to work together. I'm not saying, do so at a patient who are at late stage of cancer, probably they may have to resort to these therapy, but you have to fix your microbiome. And even oncologists have to resort to fixing, understanding their microbiome and resorting to the nutrition and therapeutics, regenerative therapeutics, apart from the toxic therapies which they are resorting to right now. Yeah.
Yeah, that's right. So, Sushant, thank you for this amazing discussion. And we will be back with another episode soon next week. Thank you so much.
Thank you.